Good morning, everybody. From the last class on synthase versus synthetase, we had left some points to be discussed later on, what we called as advanced learning concept. First was, what is source of energy in synthase reaction? Because in synthetase it is ATP. What is basis of classification? Synthetase in class 6 and synthase in class 4, although both catalyze a condensation reaction. Then there is pyrophosphate cleavage of ATP for the activation of a fatty acid by the fatty acyl coenzyme synthetase. So these three things actually I would like to discuss. First, source of energy in synthase reaction. Synthetase के अंदर तो it is clear, it is ATP, and in fact that is how we define synthetases as the energy for condensation is provided by ATP. In other words, the ATP hydrolysis drives forward the reaction or helps in the bond formation. As you can see here, a new bond is being formed and ATP is doing it. So, question comes, in synthase, as we saw, no ATP is required. So, from where the energy is coming? That's the question. Now, if you see here, acetyl-CoA. CoA means thioester. So, whenever you attach a CoA group to some compound, it becomes energized. So, it is an energized substrate which is providing energy for the forward reaction. One more example, we discussed about this enzyme of heme synthesis, ELA synthase, catalyzing condensation of the smallest amino acid with a TCA cycle intermediate. Please note it is again a CoA form, the thioester or the energized form. So ELA synthase, the condensation reaction can take place because the substrate is energized. Finally, glycogen synthase. ग्लाइकोजन सिंथेस क्या करता है? ये जैसे ग्लाइकोजन है प्री एग्जिस्टिंग, उसके अंदर न्यू ग्लूकोज रेजिड्यूज इट अटैचेस स्टेप बाय स्टेप, बट ग्लूकोज comes not like glucose. It is first energized. It is energized to form UDP glucose. It is a high energy compound, and this high energy compound can donate a glucose residue to the growing glycogen chain. So this point is clear now. Energized substrate, either in the coenzyme form or in the nucleoside sugar form, UDP glucose, so as to say. Next question, basis of classification. सिंथेटेज को हम क्लास 6 में रखते हैं, सिंथेज को क्लास 4 में. क्लास 6 में रखना, that is no problem, because class 6 are ligases which catalyze joining reaction, bond formation coupled to ATP hydrolysis, which we discussed just now, bond formation coupled to hydrolysis of ATP anyway. So that's okay. But what about these syntheses? Why they should be kept in class 4? Class 4 को अगर हम देखें, so what are these class 4s? Class 4s, they split the substrate into smaller pieces non-hydrolytically without involving any water. तो हमने तो अभी तक जो देखा था सिंथेज को as if they are catalyzing the condensation reaction, but यहाँ तो definition is different. So how do we correlate? Both are correct actually. Just to understand this, consider A plus and B. They are joining together to form a compound X. So X is the condensation product. Supposing if this reaction is reversible, in other words, if we follow the backward reaction, the reaction in another direction, backward direction. Now X is getting cleaved into A and B. So split of X into smaller pieces A and B without involving any water. So does it not become a lyase? It becomes a lyase if we follow the reaction in this direction. और जो क्लास एंजाइम की है वो तो वही रहेगी, whether you follow it in the backward direction or forward direction. So this is a lyase. Lyase is the class 4. एक और example which will make it clear to you. C6 fructose 1,6 bisphosphate is cleaved to two trioses in glycolytic sequence, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So it becomes non-hydrolytic cleavage of a compound. So it becomes a class 4 lyase. But in glucose, and this is what happens in glycolytic sequence, but what happens in gluconeogenesis? Gluconeogenesis means the reaction proceeds in opposite direction. C3 plus C3, they condense together to form fructose 1,6 bisphosphate. Because you see, the enzyme is same, aldolase. It is a reversible reaction. Aldolase can act in both the directions. So as we saw in this direction, it is class 4. So in this direction also, it should be class 4 only. So here we are seeing joining of two compounds to a larger compound without involvement of ATP, etc. So it is a lyase. So that concludes why lyases they are called. I'm sorry, the third question was about what is pyrophosphate cleavage. Pyrophosphate cleavage. So let us see pyrophosphate cleavage go. If you see ATP here is not getting converted to the usual ADP and phosphate. This is called orthophosphate cleavage of ATP. ADP plus phosphate. This is not taking place during activation of fatty acid by this enzyme, thiokinase. It is rather, instead of ADP and phosphate, it is getting converted to AMP plus pyrophosphate. Why? The question is that the answer is that this reaction requires a huge amount of energy. When ATP is converted to ADP and phosphate, the energy release is as per 7.3 kilocalories per mole. This is the amount of energy released, but this reaction requires more energy. Maybe, I don't remember exactly, maybe about 13 or 14 kilocalories. So one ATP will be insufficient. So either you use two ATPs, but our biological system has innovated a very unique way. First, ATP is converted to AMP plus pyrophosphate. And in this second coupled reaction, pyrophosphate is converted to phosphate and phosphate. More energy is released in this reaction also. So first reaction plus second reaction, both are energy releasing reaction और उनका sum हो जाता है 14.6 kilocalories, double the amount of energy which would have been obtained by this orthophosphate cleavage of ATP. So this is what we call pyrophosphate cleavage of ATP for providing double the amount of energy, which drives such endergonic reactions as this one in the forward direction. Okay, thank you so much.